When we left off in our conversation on capillary electrophoresis, we were talking about this separation right here. At left, we've got the separation of sulfonylurea drugs by HPLC. And we see that there's a lot of peak distortion, and then that GB peak is just way crazy. That's using a reverse phase carbon 18 column, which is pretty standard, and a gradient for the solvents. And that's the best you could do. It still came in at about 30 minutes, but there's a lot of peak problems like we talked about. But if we look over to the right with our CE separation, we were able to do it in less than 5 minutes, and we got better peak shape. Now, it's not perfect, right? Because we see that the first two peaks, there's a bit of coelution. So it will require some more perfecting, but the separation on the right looks quite a bit better than the separation on the left. And it's faster. So there's a lot of good options with CE, and it's got a lot of benefits. Now, we talked about why all of this was, and we talked about how that electricity, the voltage that we apply to the CE environment, is really going to motivate all of the ions in solution to move very effectively. Now, you might be saying, hey, I want to know some more details. Well, we're going to fill you in this lecture. The nitty-gritty of it is that there is a constant of proportionality between the electroosmotic velocity and the applied field, and this is called electroosmotic mobility. Every ion in solution has a particular velocity as well as a mobility. When you apply that big electric field, that voltage, capital E here, those ions are going to start to really move. Now, the degree of movement, there's a lot of things that affect that, and we talked about that in class as far as size, as well as shape, as well as charge. Put all that together, we got charge density. We're going to see how that all plays in to how mobile the electrons, the ions are. And that's really what CE is doing, is it's altering mobility of ions. Now, the apparent mobility of an ion is the sum of the electrophoretic mobility of the ion and the electroosmotic mobility of the entire solution. Think of the stampede metaphor that we talked about in our last class. The stampede is the entire solution. A particular ion would be a very particular person within that stampede of bulls in Plampona. Now, for cations, the mobility and the velocity of both the bulk solvent and the ions are going to have the same size, right? So they're positive. For anions, those are going to have opposite charges, because for the anions in the solution, remember, things are going to really want to move towards the detector. Now, what things? Well, it's going to be the cations, because the detector is by the negative electrode. So the bulk solution is going to want to move to the detector because we've set up an electroosmotic force. And for cations, the ion themselves is going to want to move in that direction. But for anions, they're going to be repelled by a negative detector, but they're going to be forced to move in that direction because of that stampede, which is the mobility of the whole solution. That's the E0 subscript. So E0 is the big stampede. EP is a particular ion. Now, at neutral of very high pHs, remember, we're deprotonating the silica wall, and we're setting up that charged double layer, which means everything's going to go towards the detector. When we keep that at neutral, meaning pH 7, which is still way above the pKa of the silica, or at even higher pHs, we're going to get a really brisk electroosmotic flow, what's also called electroosmotic transport. That's going to move those anions whether they want to go in the direction of the cathode or not. That's because the electroosmosis is faster than the electrophoresis. That's the big stampede. When you operate at those high pHs, anything higher, significantly higher than the silica, you're creating the stampede scenario and everybody goes. It's just a matter of how fast everybody goes. That's an individual kind of characteristic, but everyone's going to the detector if you've set up the EOF, that's an electroosmotic flow, if you've set that up right. So this is what we're talking about. Remember, we set up the EOF, so we deprotonated all the sides. And here's a nice diagram. It shows you that if you're neutral, you're going to be flowing with the EOF, right? Because you're not resisting it, but you're not also excited to go towards the detector. You're like neutral, right? So you're kind of going with the flow. Now, if you look at those cations, not only are they going with the stampede, remember the cations are actually attracted to the negative electrode. So that's why that observed mobility is actually a bit faster than EOF because they're highly motivated, but also they're fast because they've got that extra attraction, right, to the electrode. But if we look at the anions, they're going with the flow because EOF is so strong, but remember, they're repulsed. So they actually want to be going backwards, but look at the strength of the EOF. It's pulling them forward, but there's a lot of drag there, and that's because the negative ions are actually repelled from the detector. This is how we get that nice separation. The positive ions are coming out super fast. And again, that's going to be, we can adjust that again based on size to charge density. Your neutral stuff's going to come out all together in the middle. And then your anions that really don't want to be over by that negative detector are going to be picking up the rear. Now, at a run buffer of the pH greater than the pKa, we've got that real fast EOF. And so we're pushing all the anions forward, but they don't really like it, right? Now, if we were to drop to a low pH, our EOF would be weak and we wouldn't have a stampede. So instead of having really angry bulls in Plampona, just imagine like sleepy cows in a pasture. Like nobody's scared of those cows. Nobody's being motivated to move out of their way, right? So the anions would never reach the detector if you operated under a weak or no real EOF flow. So you want to think about it this way, right? Remember this diagram. Look at the drag, that EP, that mobility. It's going towards the positive electrode for the anions. So you look at the length of that arrow, and you look at the length of the arrow for EOF. EOF has got to be greater than the UEP for the anions, right? Because it has to overcome that resistance. Now, when we look at the positive ions, it's going to be an additive thing, right? Because the positive ions, the cations, want to go towards the negative electrode, which is where the detector is. So that's all good. They're going to go out no matter what. It's the anions that might really start to drag. So we've got to operate at a pH greater than the pKa so that we pick up all of the anions. Now remember, the neutral things here, all the neutral stuff is coming out together. So that's kind of a bummer, and we're going to have to fix that in later capillary electrophoresis techniques. Now we can control EOF. Remember, we made EOF by deprotonating the silica. So we can do the opposite. We could add other stuff on, right? So we've got this nice figure from the Harris book. You could take your bare silica, right? So you can see that as we increase pH, our EOF goes up, and electroosmotic mobility, remember that's the actual velocity, the mobility per unit area for voltage. So that's your meter squared voltage inverse and second inverse. That's your EOF right there. So we can see how with increasing pH, bare silica, then our mobility goes up. But if we want to control that, we certainly can. We could potentially slap a positive cationic polymer that would reverse the EOF. That is on the bottom right. So we could do that. And that would mean that we would have the detector going the other way, which is easy to do. We could just swap out the electrodes. But that would mean that our negative things would come out first, our cations would come out last, and our neutral stuff, though, would somehow still be in a clump in the middle. Now, this just depends on what we're really interested in. Are we interested in the items that are negative, or are we interested in the items that are positive? So which analytes are the most interesting to us? And by kind of reversing the EOF, we might get a completely different, right? We are going to get a different elution order. And so that might be what we're going for. We could also stabilize the EOF by adding what's called a negative dextran sulfate, which is basically a fancy way for saying a negative ion soap, which would stick to the walls. So what we would do with this is that we wouldn't operate under a high pH. So we wouldn't strip the silica off. What we would do is run it under a below the pKa, but we would apply this anionic dextran sulfate, which would stick to the cyano walls because they would have this dipole, right, with this hydrogen bonding. So that dipole ion interaction there, you'd still get like a reversible coating on the silica wall. It's not as extreme of an EOF. It kind of stabilizes it over a wide pH range. And so we might want to do that if we're interested in very particular analytes. So remember, we make EOF so we can modulate it at will. That gives us a lot of power when it comes to CE. Now, we've got to think about a couple options because with CE, we do have some different sample injection options. We can do good old-fashioned pressure injection, and this is pretty popular. We can calculate exactly the amount of volume injected using this equation here. Don't worry about memorizing it, just showing it to you, that we can actually calculate the exact volume down to microliters and sub-microliters. And so this is going to be about the viscosity of the solution, the diameter of the capillary, the time that we're actually putting it under pressure. So that, pi is in there because, again, it's a cylinder for some unit area, under some level of pressure, for some amount of time, right? And so we can calculate that. The other way you can inject is, remember, this is all under ionic control. We got a lot of charged species in solution. So you could use electrokinetic injection, which is like, boom, shock that sample solution and have it move towards the capillary. The pressure is actually more common because you just get a uniform plug that's not already doing weird things with the charges. But it all depends on what in particular you're separating. So there could be a reason, if it's a real complicated ion solution, that you already want to kind of influence how things are separating with injection. So we can calculate the moles applied using, again, some of these terms here. Again, don't worry about memorizing this equation. Just see that the field strength that we apply is directly proportional to the moles we inject. Now, what are we going to do about some stacking? Because you might have remembered we were like, yay, our peak shape is better, awesome. Well, there's a limit to that. We have to think about that. So just like in our reverse phase liquid chromatography, we had those sample effects where if you overload the column right, you got skewed peaks. If you have some kind of weird ghost peak behavior, you'll have secret second add-ons, right? And then you've got some weird stuff. Maybe your column has just got those patches of differential stationary phase. So you start getting some real kinky stuff. So you can have the same issues in CE, but they're caused for different reasons. They're caused because of conductivity differences. Now, stacking is a very particular strategy used to focus analytes into narrow bands at the start of a capillary. What we're going to do is we want the sample plus the electric field to be greater than the electric field of the background electrolyte. You have ions in a sample plug. Then they're going to reach this boundary. And this is what's going to happen. So what you're doing here intentionally is that your sample is a higher ionic strength, so that when you apply the voltage across it, it has a higher conductivity. Then it hits the background buffer, which is just in the column. And because there is this boundary and this difference, all the ions actually, boom, kind of come to a stop. They don't really stop. There aren't little brakes on ions, but they slow way down because there's such a difference in conductivity between the two solutions. That means that they kind of stop at this liquid boundary or slow down, which stacks them all top of each other and keeps them in a really tight band. Stacking will continue until most of the analyte cations are concentrated at one end of the sample plug and anions are at the other. So your sample you've made it super salty, essentially, and then you're injecting it into a less salty but bigger volume capillary. And in that zone, they're going to separate out by cations versus anions, because remember, the detector is negative. So the positive things are going to want to kind of spread out in front. But they're going to be stacked right on top of each other in a fairly tight band. So here's a good visualization from Harris. Okay. So what you have here is you've got your high electric field, so you got a lot of ions, but you also have a lot going on in your background buffer. It's highly conductive because it is in a buffer. Your sample plug has low conductivity. Your background buffer is higher concentrated, but a really high electric field because you've scared the crap out of it basically to get it into the capillary. And the differences between the two mean you have this stacking going on. Now, we can see what the stacking does. It actually makes those really nice, super narrow peaks. So these are incredibly narrow peaks instead of having that broadness. So it's a way for us to limit the broadness and get everything really tight and concentrated so that we get that even more extreme symmetry about an average centroid value. It's a really easy trick. You basically make your sample buffer at a different concentration than your background buffer. Easy peasy. But of course, you can go too far. And if you go too far, you'll get things like this. You want a difference between your analyte solution and your background, but it can't be too much of a difference, right? There is such a thing as too much of a good thing. This is what happens when you've gone too far. Now, these shapes should look familiar. It looks like fronting and tailing. Now, the difference here is the cause of the fronting and the tailing. If you've got these types of shapes, you in CE, you know exactly what happened. You just went a little bit too far with the differences between the sample solution and your background buffer. Now, CE, as we've seen thus far, has no stationary phase. It's just that bare silica, some buffer in solution, and some ions. Well, that's not really true, because there is a particular type of CE that does include a stationary phase. Sort of. It's called micellar electrokinetic chromatography, or MEKC for short. This is the one I talked about where we add soap. Remember, soap is just something with a hydrophobic tail and a hydrophilic tail, and then they assemble because the things that are hydrophobic don't want to exclude water, right? They don't want to be around water. And the things that love water are going to assemble so that the water-hating stuff is on the inside, the water-loving stuff is on the outside. And what you end up getting is this micelle. Now, this is great because stuff that also doesn't like to be around water can float into and partition into the interior portions of the micelle, and things that, like water, can stick to the surface. Well, this means that the micelles are acting as a pseudo-stationary phase. One of the most popular surfactants to use is sodium dodecyl sulfate, abbreviation SDS. You probably heard this before in not only our class, but in biochemistry, because this is the soap that's added to SDS page, a particular type of electrophoresis. What we can do now is we can separate neutral analytes as well as ions, because the neutral stuff can get hidden in these particular micelles, but the degree of which they'll intercalate themselves or insert themselves into the micelles is going to depend on their structure. It's going to be just like a stationary phase in our reverse phase chromatography. Of course, the one caveat is that the surface is covered with these charged hydrophilic heads, like sulfate. So that means that charged things could stick to the surface or be repelled. So it's a pretty complicated pseudostationary phase, actually. But for a particular pseudostationary phase, we can map that behavior really, really well and get a really well-defined elution order. So here's the whole thing in action. So this is an SDS surfactant being used as micelles. So the whole micelle is negative because of that sulfate group. And now you can have partitioning into and out of the micelles, just like a stationary phase. Neutral molecules, positively charged ones, negatively charged ones are going to be like, no way, no thank you, right? So you've got that differential separation, which is going to be great because that's by definition chromatography. Our electrophoretic migration of the negative micelles, if you use a negative surfactant, is to the left. Remember, they're all attracted to the left, right? But remember, EOF is going to push them all to the detector. So even though the micelles are repulsed by the negative electrode by the detector, the EOF will push them that way. But the micelles will kind of drag a bit. So even though the pseudostationary phase is moving, it's not moving as fast as the bulk mobile phase. So that's why we call it a pseudostationary phase, because it's more stationary than the mobile phase. And that drag or that slowness allows the micelles and the analytes to interact and for the differential separation to occur. Now, in the absence of negative micelles, remember, all the neutral stuff would come out at the detector at the same time. They're not coming out with the cations, they're not coming out with the anions, they're all dumping together. Now, if you add in anionic micelles, they'll be injected and reach the detector at the time that's called TMC. Remember, anionic micelles are going to be repulsed by the detector, but they're eventually going to get there because of the EOF. But the TMC time, which is basically the time the stationary phase is eluting from the column, is going to be way longer than the void time, the T0 time. So you're going to have plenty of time to get your differential partitioning between your ions and your neutrals and your micelles. If a neutral molecule equilibrates between the free solution and the inside of the micelle, its migration time is increased, right? Because remember, the negative micelle is lagging behind because it is repulsed by the electrode at the detector. So if a neutral thing is hanging out with the micelle, it too gets slowed down. It's like a relay. The slowest member of the relay determines the overall pace. So if relay runner decides to chill and run with the third person who's super slow, 3 and 4 both become slow. That's what you want to think about here. Now remember, we've got mass transfer. The mass transfer term is no longer 0. See, that's the benefit of when it was just CE with no pseudostationary phase. The Van Diemter was real real nice. And that's why the peaks looked the way they did. You had diffusion, but you didn't have multiple paths. And mass transfer was not an issue because things weren't partitioning because there was no stationary phase. But if you add micelles in, aha, you do have the mass transfer term. Now, what you don't have are the multiple paths because it's not a solid packed column. But mass transfer is back, and that means we're going to have a little bit of Van Deemter drama. The good news is that the band broadening is modest because the mass transfer is still very small, and what we need to remember is that the stationary phase is still moving. Everything's under this tremendous EOF. So the mass transfer is just enough to help us with our neutral compounds, but not too much to significantly broaden our peaks. So we're not going to freak out just yet. Now we can use all kinds of additives, anionic surfactants, cationic surfactants. We could even use neutral surfactants. We could use chiral inclusion molecules, polymers, all sorts of stuff. A real popular additive are these chiral inclusion complexes that are called cyclodextrins. They vary in size, so you kind of can think of these as not only being chiral, but also being a bit size exclusion, which is really interesting. And these are used quite a bit in CE and MEKC. So again, we can use chiral micelles or, like those inclusion complexes, chiral inclusion complexes to separate out column molecules. Look at this beautiful chiral separation that we've got here from the Harris book. You've been able to separate out these chiral compounds in about an hour. It's awesome. And because you're just adding in, you're sprinkling in a soupçon of these chiral compounds into your background buffer, you don't have to have any kind of expensive specialty columns that you're going to have to regenerate because they're proteins, blah, blah, blah. Nope. So doing chiral separations on CE is actually cheaper and easier and faster. Now, another cool thing we can do, which again, if you've been in biochemistry and advanced biology courses, you probably know a lot about, is our capillary gel electrophoresis. So again, this gets us back into our SDS page territory. So we have capillary gel electrophoresis, which uses physical gels of entangled polymers. And what this is going to do is you've got this covalent cross-linking. So this is kind of making one big gel sieve. So macromolecules are going to be separated by sieving, which is when the small molecules can slip through, right, these holes, and they get to the detector first. And the big molecules are going to get kind of caught up and entangled, and they're going to elute last. What you want to think about is that this is like reverse size exclusion chromatography, but really fast. Because remember, you're not only having them go through under pressure, you're also going to apply a huge voltage behind these things. So it's really fast reverse size exclusion chromatography. This is why SDS page is so popular in biology and biochemistry. It's got all the benefits of size exclusion with an additional benefit of being way faster. Now, again, the difference here is that the small things can slip through, right? Because it's this massive, you want to imagine it like Jell-O, but Jell-O with spaghetti, just a huge amount of spaghetti. And the big molecules get tangled up in the spaghetti, but the small molecules can slip through the spaghetti. So the small molecules come out first, the big molecules come out second. So that's why we say it's reverse size exclusion. But everyone's under, say, 10, 15, 20 kilovolts. So when we say slow, we really mean slower. And this is how we do DNA profiling. It used to be that slab gel, right? Pour it out, do it the old school way. You had little bars, and you can still see those images. Classic SDS page. But how we do it nowadays is the gel is in a capillary. You inject it into the capillary first, it fills up the space, and then you're going to inject in a small sample plug. And you're going to get a spectra that looks like this. Because oftentimes you've added on a fluorescent tag, so you're looking at really a fluorescence detection here. Now these peaks should look real familiar because you're looking at an electropherogram. So it's a chromatogram, right? But we call it electropherogram because it's for electrophoresis. But this is how we take advantage of all the power of separating out these fragments. So what you've got here are, quote, non-coding regions of DNA, usually 12 to 13 to 15 specific regions that are called loci. And everyone's got variety of versions of these that they've inherited from their parents. And so the combination of all this data together can be used to identify individuals. So what you're really looking at is those sections, right, that correspond to those particular numbers. Then every single one that you've inherited that produces this value or this specific sequence is going to have a different elution time because they're different pieces. And so we go through and we clip out all these pieces and make copies of them, say through PCA. And then because they're charged, these are, right, big nucleotides, then we're going to separate them also based on size because they're different lengths. Remember, different sections of DNA and have the different lengths for different coding regions. And so they're going to be different sizes. They're going to be different charges, and put those together, they're a different size to charge ratio. They're going to have different mobilities when we hit them, right, with this electricity. Now, because they're different charges and different sizes, we can use that gel electrophoresis to separate them out and get different elution times. And the elution times will correspond to different particular loci, and that's how we get these profiles. Now, I have really summarized it way down. It is more complicated than that, but you're still separating out those nucleotides by size and by charge. This is one of the most powerful applications in the last 30 years of this type of chromatography. In fact, this year is the 30th anniversary of the first criminal case involving forensic DNA profiling. Now, lab on a chip is just an extension of CE, but it's also a way that we can speed up the whole process. We can use glass or plastic chips, just like we would a computer chip, to employ electroosmosis or pressure. These are such small volumes, it's really easy to make this work. We're going to move liquids through sub-micron size channels, right? Teeny tiny. Again, think computer chip size. Now, why do we want to do this? Well, because if we can do it smaller, we can do it faster. Because smaller volume, smaller length of the channel, things would move a lot quicker and we could get done in a few hours. With a dedicated instrument analyst, conventional DNA analysis can be done in less than 5 hours. I think actually right now we're pushing about 4 and a little bit less. But with better automation, we can get that down to 2 and 3. Lab on a chip has the potential of being less than an hour. Some folks are aiming for less than 30 minutes. Now, this doesn't include replication, but it does include the analysis of a known. That's wild, but we're almost there. Here's what a chip might look like from our textbook. And PCR, which is how we're basically going to photocopy genetic code, okay, so that we can amplify it so that we can do this type of work. Everything is literally all in the chip. From your PCR to your separation, everything's there. Look, you even have a waste reservoir. This can absolutely all be fabricated. Again, if we can fabricate really complicated computing chips, we can fabricate this. The key thing is is that we've got to get it to work reproducibly and get that cost point down. We're not quite there on both of those fronts yet, but we are getting extraordinarily close. Here's one great article, and I've got the link to the paper that shows you how close we're getting. We're using all kinds of great innovation and fabrication. Some of the limitations are due to materials, our ability to reproducibly manufacture these things. We can do it with the computers, but we're going to need to use different materials because remember, we're going to have to apply pretty high voltage. We're going to have to do, like with PCR, particular chemical reactions in designated reservoirs. We've got fluid movement. That's a bit different, right, than a computer chip. So while we can do the fabrication at this size and level and detail, the challenge is that we're trying to do it. It's also solution-based. We've also got to include detectors in here that are optical. These are where the challenges are. But the advancements we've made in the last 5 years are mind-blowing. Now, again, the advantages in summary to capillary electrophoresis over conventional chromatography, so imagine our reverse phase, is that as we've seen, we have much higher resolution. Remember, we're using teeny amounts, so low waste production. I did years of CE in graduate school, and I also did a lot of HPLC. You've seen how much liquid waste we can go through in HPLC. In CE, no joke, I did both methods, right? HPLC and CE for 4 solid years in graduate school. In 4 years of using capillary electrophoresis, and I did MEKC, I didn't even produce the same amount of waste as 1 month in HPLC. It's wild. Now, the flip side there always is one. We have low waste production, but CE is not for preparatory scale type stuff. It's really strictly for analytical analysis and those really small quantities. Now CE, they say simpler equipment. Yeah, okay, I'll give it to Harris on some things. We don't have some of the complicated pump systems and things like that. But you've got a system under tremendously high voltage. So I kind of put this one down because Harris does, but I got a bit of a side eye on it because when you have something under tremendous voltage and you're applying it over some unit area, you've got to really maintain that system, right? So I would actually, as someone who used side-by-side instruments for years, I would kind of call it a draw. Each one has their unique costs, their unique problems, and their unique maintenance requirements. Now, some drawbacks. We're going to have higher limits of detection, and that's simply because of the detectors we typically use. But if you do fluorescence, you can get right back down to that scale. Now, your run-to-run reproducibility is an issue because of literally voltage and electronics. Sometimes you can have surges. You've really, again, got to maintain that. But this is why you always include a migration time marker so that you can correct those things. Now, it's a drawback, but remember that in reverse phase, you also had to include a marker, right? You had a void time marker so that you could reset and actually use the TR or the corrected retention time. So everything's got that drawback. Now, what you might have for some analytes is an insolubility with some common electrolyte solutions. However, we can fix that by adding micelles, which, as we know, soap is good at dissolving a lot of stuff. So it's a drawback, but we could switch over to a different type of capillary electrophoresis and fix that issue. Again, the biggest drawback, one of the biggest, is our inability to scale it up. This is really limited because of our power consumption, right? You're not going to want to scale it up to preparatory size because imagine how massive your electric field would have to be. We already have energy consumption issues. This would just really be bananas. not when we've got other larger preparatory scale, liquid phase, conventional chromatography we can use that works great. We don't want to have to introduce a whole other power grid so we can make this other type of chromatography preparative scale. It would be cost prohibitive. Now detectors, here's our options. You've got your classic UV, and this is actually going to be on column. This is why you get some sensitivity issues, because you're going to have to actually burn an optical window through the capillary. So the capillary glass part's going to be intact, which is completely transparent to the detector, but you're going to actually burn off the plastic coating. So remember, path length and absorbance are directly proportional. You're not going to actually make that big of a window. You're going to make it in the micron range. Well, you can see already what that does to absorbance. What it also means is that because it's a flow-through system, like in our other types where we've looked at solutions, your background's got to have like little to no absorption in the range that you're looking at. Now, borate buffers are really popular here because borates are transparent over most of the UV visible range. So we've got to think about that, but we already considered that when we did spectroscopy in our conventional UV. You're not going to use a solvent that also absorbs. So we've got to keep that in mind here too. You can use fluorescence detection, and this is where you can deal with that other criticism of, oh, it's not as sensitive, or the LOD is too low. Hey, if you can switch to fluorescence, which is how all forensic DNA is done because all of that's fluorescently tagged, you can really dramatically shift this to a dynamic range of 10 to the 9th and take care of that whole criticism. It's pretty standard now. Again, it's absolutely standard if you're doing any kind of forensic analysis to have a fluorescence detection. And these detectors have gotten cheaper and cheaper because you're able to get this massive dynamic range, and it's just considered more standard. Even when you have to pay for it to get the additional fluorescence detector because UVs are still considered standard, the UV vises, it's absolutely worth the money because it's going to extend your range. It's going to really deal with your sensitivity issues. Now, this is an electrochemical method, so we can use all kinds of electrochemical detection schemes like conductivity detectors. This is how we can often get things to be chip-sized. So it's awesome. Same type of conductivity detector as we used in GC. We're going to measure the background of just kind of the background electrolyte, and then we should see a difference between the background electrolyte and when an analyte is passing by. We can use amphomeric detection. Again, any of your electrochemical detection schemes that can be used in general can be merged with CE because we're already doing electrochemistry. And probably the biggest and most important is that we can merge CE with mass spec. It's awesome. And the good news is that we merge it with a very particular type called electrospray mass spectrometry. So electrospray mass spectrometry, as we'll learn, is going to use this nice electricity voltage separation to actually make a nice uniform spray and get these tiny droplets that are charged for mass spectrometry. If that sounds like it's a match made in heaven with CE, it absolutely is. It's a dream to work with one of these instruments. So mass spectrometry is going to be our next section after the next exam. So we'll be jumping into MS, which we've gotten some familiarity with in lab, but we'll be dealing with some real practical things when we meet next time for lecture, which will be on Wednesday of next week. Remember, Monday is the exam. Multiple choice as per usual. Thanks, everybody, and I hope you have a good weekend.